In this video, I'm going to talk about the main types of organic molecules. There are four main types: lipids, proteins, carbohydrates, and nucleic acids. First, I'm going to talk about lipids. Lipids are hydrophobic. Hydro meaning water, phobic meaning they don't like it. So you can think like oil on water, how they separate out very quickly. This also implies that they won't dissolve in water. Lipids are energy rich, meaning that for not a whole lot of weight, you get a whole lot of energy. Many lipids are made of fatty acids, which in and of itself is a type of lipid. But fatty acids are building blocks for other types of lipids. There are lots of different types of lipids, and I show a few here, including fatty acids, triglycerides, phospholipids, and sterols. I mention these in particular because they're important biologically. Okay, so the first type of lipid, fatty acids. You will either have saturated or unsaturated fatty acids. But first let's talk about fatty acids in general. They have this carboxyl group at one end, and then a whole tail of hydrocarbons. Call it hydrocarbons because it's just hydrogen and carbons, which you can see here. There's just a ton of carbons with hydrogen sticking off, and that's all a fatty acid is. This carboxyl group here, and then a long string of hydrocarbons. So back to the saturated, unsaturated thing. If it is saturated, as in this one here, there are no double bonds. Versus unsaturated, you can see the double bond here in red. The double bonds cause a kink. So you can see in this one we have a bunch of yellow and blue fatty acids, where the blue dot is this carboxyl group, and the yellow is what the carbons are doing. When they're saturated, when they don't have any kinks, they line up very nicely. If they're unsaturated, like a lot of these are, not all of them, but the kinked ones are unsaturated, and they don't line up as nicely. So it causes them to have different properties, like when they kink and push each other apart, they don't want to solidify. So like butter or something that gets solid when it sits on your countertop will be more like this, versus oil that won't solidify is more like this, because all the molecules are pushing each other apart and not wanting to become solid like this. I just want to show you one more picture. This is another way you can write it. You can see the saturated with no double bonds, and the unsaturated with that little double bond spot right there. And as far as the word saturated goes, it's saturated with hydrogen. So in order to make that double bond, we had to take out some hydrogen. So that's actually what the word saturated is referring to. Okay, the next type of lipid, triglycerides. They are made of glycerol, type of molecule, and then 3 fatty acids. Triglycerides are a way your body uses to store extra energy. In general, like I said, glycerol, which is this red thing, and 3 fatty acids. You can see one fatty acid chain. Each of these chains with in black is a separate fatty acid. There are 2 main types of triglycerides. When they're made of all unsaturated fatty acids, when they're made with any unsaturated fatty acids, we call it an oil. Like over here, see how there's some double bonds, some kinks in the fatty acids that were used? That causes it to push other molecules apart and to become more of an oil. when it's made with all saturated fatty acids. Remember, saturated means straight, no double bonds, no kinks. It's stored as a fat. So like this, in this picture here, you can see how these are all straight, no double bonds, no kinks. Therefore, those are all saturated, and they can sit there very nicely together without pushing each other apart. The next type of lipid is phospholipids. This is a very important part of what cell membranes are made out of. In general, we say they have a hydrophobic tail. Remember, hydrophobic meaning they don't like the water. And hydrophilic head. So, you can see sort of a picture here of a cell membrane. And what happens is all these little yellow squiggly things are tails. And the tails, since they are hydrophobic and they don't want to touch the water that's out here, they all come together and put the hydrophilic heads, remember hydrophilic, they like the water, so the hydrophilic heads face the water and protect the hydrophobic tails, which don't want to see the water, don't want to touch it. So when you have this, it's called bilayer, bi because you have one layer here, one layer here, it allows it to sort of accommodate what the head and what the tail like. And that's what your cell membranes do. That's what your cell membranes are made out of. For an individual phospholipid, here's this blue dot is the hydrophilic head, and these black lines are the tail. Remember, there are two little hydrocarbons that are the tail. So here's a more detailed picture. You can see this spot right here is sort of your glycerol, and then your hydrophilic head here, and then these sticking out here are your hydrophobic tail. And then this whole thing is also part of your hydrophilic head. So to say that again, this is your hydrophobic head, and these are your hydrophobic tail pits. The next type of lipid is called a sterol. They are made of 4 hydrocarbon rings fused together, like you can see here in red. These rings are all hydrocarbons, you can tell, because if there were anything but hydrogen and carbon, they would have to write it. Like down here, that O for an oxygen, you have to actually write that. But these rings, 1, 2, 3, 4 rings fused together, so they're all stuck together on one side, form a sterol. In this particular case, we have cholesterol. And you can see there's some other stuff sticking off. The CH3 here, CH3, lots of other CHs here, and OH here. But that's just what makes it a particular sterol. To be any type of sterol, it just has to have this. Cholesterol is the one we have here, but also steroids and vitamin D would be examples of sterols. Next, we have carbohydrates. Basically, carbohydrates are sugar in many forms. They are made of carbon, hydrogen, and oxygen, and there are 2 main types: simple sugars and complex sugars, or you could say simple carbohydrates and complex carbohydrates. First, I'm going to talk about the simple sugars. They are used for short-term energy and are the monosaccharides and disaccharides. Note that one sugar, a monosaccharide, is well, just vaguely, it is some sort of ring with oxygen and some other stuff sticking off of it. So just as a general visual recognition, here we have glucose, a monosaccharide. It's a ring with, you can see, some oxygen and other stuff sticking off of it. Here we have another monosaccharide, fructose. Here we have a disaccharide, so two monosaccharides stuck together, making sucrose. Alright, next we have the complex sugars. These are for long-term energy. They are made up of lots and lots of monosaccharides all strung together. When we string monomers together, we call it a polymer. So in this case, they're called polysaccharides. Poly meaning many, saccharides meaning sugar. So we have polysaccharides are lots of monosaccharides together. We have some examples here. You can sort of see the ring with stuff like starch, cellulose, glycogen, and you can see how they're a little bit different from each other, but they're still essentially monosaccharides all strung together in some way. Next, we have nucleic acids. First, what do? Well, one thing they can do is store genetic information, like DNA does. They can also transmit genetic information, like RNA. Next, what are they made of? They are made of strings of, or remember, polymers. That's what we call it, polymers of nucleotides. So nucleic acids are made of nucleotides. What are nucleotides? They are essentially three things. A nitrogenous base. We say nitrogenous just meaning it has nitrogen in it. We have sugar, which in particular will be ribose or deoxyribose, pretty much. And phosphate, a phosphate group. Just a general note about this ribose and deoxyribose. Think DNA. What does DNA stand for? Deoxyribonucleic acid, right? So DNA is just specifying that this is the type of sugar we're using for that nucleic acid. Deoxyribonucleic acid, DNA, versus RNA, ribonucleic acid. We have ribose instead of deoxyribose. But here we have a picture, the nitrogenous base, in pink. And over here we have examples of different possible nitrogenous bases attached to a sugar. This sugar can either be ribose or deoxyribose, and our phosphate group in yellow. And when you take a bunch of these and string them all together, you can make DNA or RNA. Next, we have proteins. What are they? Well, they're made of nitrogen, carbon, hydrogen, and oxygen. Nitrogen being a particularly important thing. More specifically, they're all made of amino acids that are covalently bonded together to make a polypeptide chain. So, you take amino acids, you stick them all together, and you make a polypeptide chain, and that is what protein is made out of. It's made in a process called protein synthesis, easy enough to remember. And in general, they're categorized by what they do. So if they're used for storage or structure, I put it in quotes because they all have a structure, but what people mean when they say used for structure is like sort of your structural support. like in a house, you have concrete blocks or whatever you have to support it. Well, the protein can do the same thing in the cell. They can also be used for transportation. There are lots of molecules that, or lots of proteins that sit on the cell membrane and make sure things get in and out of the cell as they want them to, or as the cell needs. And they're also used for catalysis, like enzymes. For example, we have enzymes, we have hormones, we have keratin. I put sometimes next to hormones because there are proteins that are made of, hormones that are made of proteins, but a lot, if not most, hormones are made out of steroids, or sterols, which is a type of lipid. Alright, so next, the amino acids. We know that proteins are made of amino acids, but we have to know what an amino acid is to understand this all the way. There are 20 different common amino acids. They all share a general structure, like you can see this big picture here. They have an amino group in green here, and an acid group. This is a carboxylic acid, so we have amino acid. Now, what makes them different from each other, the 20 different amino acids different, is this R here. Now, in chemistry, R just means this could be anything, basically. For example, over here, we have glycine, which has an H there, or methionine, we have this thing here. For lysine, we have something different. For phenylalanine, we have something different. But in this R position, you can have a lot of different stuff. And that different stuff is what makes the amino acids different. They have the rest of this green, gray, blue in common. The R is what's different between them and what makes them special. So, one very, very, very important thing about proteins is that structure equals function. There are 4 different levels of structure: primary, secondary, tertiary, and quaternary. Primary structure is the amino acid sequence. So, for example, if you have methionine, arginine, lysine, cysteine, that'll be a different protein than something that starts with methionine, tyrosine, phenylalanine, cysteine. Primary structure is the amino acid sequence like you see here. Only proteins have way, way more amino acids than that, but this is just a sample. This amino acid sequence is coded for in the DNA. So the DNA is telling which amino acids and what order to use. This primary structure, this sequence, is what decides just about everything about the protein. Remember, structure equals function. So this is the beginning of the structure. So it's the beginning of the function. The secondary structure. So you have a string of amino acids, and they tend to want to do 2 things very commonly. They want to create alpha helices, like you see here, this red and green. These loop-de-loops are called alpha helices. And they want to make what are called beta pleated sheets, sometimes just called beta sheets or pleated sheets. On the right here, though, you see it, where that's more of a flat sheet. You can see some alpha helices behind them, but the colored here is the alpha helices, and here's the beta pleated sheets. These are 2 common things that your strings of amino acids want to do. It's called secondary structure. Next, we have tertiary structure. So this is how it folds overall. In other words, it's how the alpha helices and beta sheets fold onto themselves. So you have a big string of amino acids, which then loops itself around and makes some alpha helices and beta pleated sheets. So then you've got a big string of alpha helices and beta pleated sheets. Well, the tertiary structure is when those things sort of come in and all stick together on top of themselves, like you see here. Lastly, we have quaternary structure. When you had this tertiary structure, it was all the same sequence of amino acids. And what I mean by that is all the amino acids in this polypeptide chain were connected. It was one polypeptide chain. When you have quaternary structure, you get multiple polypeptide strands. So picture a couple of these tertiary strands. You have the tertiary strand of this string and the tertiary strand, or the tertiary structure of a different string, come together. Like in this picture, you can see each of these colors is a different, was a different polypeptide strand, and they all come together and stick together. Each individual polypeptide is called a subunit. I say they may be same or different. These all sort of look like they were the same sequence, so they all had the same primary structure initially. So we have 4 of the same primary structure, but you don't have to. Sometimes you can have quaternary structure where 2 different things will come and stick together. But they do this for function. All the subunits are needed for function. Now, sometimes you have lots of subunits, sometimes you don't. But however many that protein needs, you need all those subunits for function. So the quaternary structure is when you have multiple polypeptides come together to make a functional protein. So, in summary, I put structure equals shape equals function. The DNA codes for the amino acids. The amino acids make a particular shape. And the shape is what makes it work in its particular job, whatever it's supposed to do. So, if you were to take away the shape, you would take away the function. It would not work anymore. This is called denaturation. It can be caused by heat, by salt, by a bad pH. For example, if your blood needs to stay near neutral or near 7, and all of a sudden something happens and it drops or it goes up, either one. That's bad, that's out of the norm, and it can denature proteins. So here you have sort of a squashed picture, but you can see it anyway. How you had the normal folded functional protein, because this structure is the function. It can do the function. But when you lose that structure, when you denature that protein, you lose the function as well. And you can put it back. In general, proteins can be put back if you just take away the heat, or take away the salt, or put the pH back where it should be. But it's very, very important to note that the protein structure equals function. I'd say that's probably the most important single fact you should understand about proteins overall. Structure will equal the shape, which equals the function.